Okay, let's welcome our next speaker, Dr. Ahrens. He is a senior group leader at Howard Hughes Medical Institute, and he studies how large neuronal populations correlate to enable flexible behavior in animals. Today, he will give us a talk regarding to whole brain neuroscience using the larval zebrafish as a model organism. Let's welcome Dr. Ahrens.
Hi, everyone. Thank you for the introduction, and thank you to the International Congress of Basic Science for hosting me, and also thanks to the organization and the people who nominated our paper for the Frontiers of Basic Science Award. It's a great pleasure to be here. And I'm very grateful for the opportunity. So I'd like to talk about whole brain neuroscience in larval zebrafish. I'd first like to acknowledge the work that was selected for the award. It's called Whole Brain Functional Imaging at Cellular Resolution Using Light Sheet Microscopy within biophotonics. And this was work with Michael Orgar at the Champalimaud Center for the Unknown, Drew Robson, and Jennifer Lee at Max Planck Institute, and Philip Keller, also a senior group leader at Janelia Research Campus, with also thanks for support by Florian Engert and Alex Scheer. I'm going to go over that work in this talk, but I'll also discuss more recent work by the people on this slide and collaborators, especially work by N. Young. And then if there's time, we can get to work by Yumu. N. Young is now a professor at UNC and Yumu at ION in Shanghai. So to motivate kind of our general research program, which is really based on interrogating the nervous system at the whole brain scale, I just want to give an example of animals that we're not studying, but that make a useful point. So these are gazelles and African wild dogs. The dogs are hunting the gazelles, but the gazelles have a way to hide from the dogs using their special abilities to stand on these very steep cliffs. The dogs try to repeatedly get to the gazelles, and presumably in the hope of attacking and eating them. But they find out they cannot do it, and eventually they give up. So they stop trying, and this is a very kind of normal. It's very intuitive for us, this kind of behavior, and if you break it down, you see that there are many components of this. So presumably the dogs initially had a sense that they're hungry. They saw the gazelles. Then they assessed the environment, the geometry of the environment. They learned from their previous attempts of getting to the gazelles and failing. And then eventually they decide to abandon the hunt. And in neuroscience, what's historically often happened is that people isolate one of those components and study it in great detail in a specific brain area that may be dedicated to that function. For example, the olfactory system is specialized in detecting the smell of the prey. And so you can learn a lot from studying the olfactory system and see how neurons in the olfactory system respond to the smell of gazelles versus the smell of fruit, for example. But if you consider one of those behaviors in their totality, you find that all kinds of systems are involved: sensation, decisions, memory, and action. And so we also know that all these functions occur in different areas of the brain. So this is the brain of a dog, I believe. And we know that, for example, one of those areas, the cerebellum, is involved in motor control, so in movement and learning how to move more accurately. There's visual cortex that's specialized in processing visual scenery, and so on. And it's very difficult in large animals to study all these brain areas at the same time because it's difficult to get to the different parts of the brain because the brain is so big. But that poses a challenge because, as I said, we know that neural processing is distributed, that any one function engages neurons across the brain in many different brain areas that work together through the dense interconnectivity between all these neurons. So computations occur across the brain. Many brain areas interact to produce a single behavior, and we don't always know at the start which brain area to even look at. There are many cases where the animal does something and we have no idea what part of the brain is responsible for it. And also when animals learn, these connections between neurons can change in strength. That's one, just like artificial neural networks, the weights change, and that's often an implementation of learning. And these weight changes can also occur between many neurons in different brain areas. And so plasticity is also often distributed across brain areas. So these are all reasons to study the brain at the whole brain scale and to find techniques to expand from studying small areas of the brain or small numbers, small fractional numbers of the brain, to studying almost all neurons in the brain. And so in mouse, we compare to zebrafish, the mouse brain is much larger. So the mouse is a very useful model for neuroscience. It's been widely studied. The behavior is quite well known, although all these animals always continue to surprise us in how smart they are. The more we study them, the smarter they become. But the mouse is also challenging from that whole brain perspective. It's got 70 million neurons, and at the moment we can record from about 100,000 using very advanced techniques, 1 million of those neurons, but that's still between 0.2 and 1% of the entire brain. Now in zebrafish, because they're transparent and we can use microscopy techniques to access the nervous system, and because the brain is much, much smaller, it's got about 120,000 neurons, we can record from about 90% of all neurons. So it's possible through optical techniques to get a quite comprehensive picture in terms of numbers of neurons of what the entire brain is doing. And so that's been our research program to really exploit that in larval zebrafish. So this is a fish that you can buy in a pet store. It's a few centimeters big, but when they're 1 week old, they're tiny. They're 4 millimeters long. And at this age, they already perform a variety of very interesting behaviors. They hunt, so they watch prey in the environment. They make decisions about which prey to try and eat. They can perform motor learning. They can learn things are dangerous in the environment or things are safe. And this is kind of remarkable because these are only 1 week old. So that means they're 1 week after the single cell stage. They develop outside of their mother. So 1 week ago, this animal was 1 cell. Now it's a complete animal that can perform all kinds of interesting behaviors. And we can genetically manipulate these animals to express, for example, calcium sensors in their brain. You can see this animal on the right-hand side. You see 2 halves of the brain. The right-hand side is a fluorescence image where we excite the green fluorescent protein with blue light. And on the right side, you see a single plane of that image where you can see individual neurons. When these neurons activate, calcium goes into the neurons from the extracellular space. That calcium causes the green fluorescent protein to become brighter. So this tool is called GCAMP. And if we look at the brain anatomically, we can label excitatory neurons and inhibitory neurons in different colors. So neurons that cause other neurons to spike, or neurons that cause other neurons to spike less. In different colors, you can see that on the right. And what's important is that this is a vertebrate animal. So many of its brain areas are shared with humans. It's got a habenula, a cerebellum, inferior olive. It also has a very evolutionarily early version of cortex and hippocampus and the brainstem. So we can make a lot of analogies between humans and these tiny creatures, although we always have to be careful not to take the parallels too seriously. Now, the way we can record from this brain at high temporal and spatial resolution is through light sheet microscopy. And the principle here is, if you see my poor rendering of a three-dimensional brain, you can imagine shining a laser beam through it, but the very miniaturized laser beam, and scanning it plane by plane and building up a picture of the entire brain within a fraction of a second, and then repeat that for the next time point. And in this way, we can build up a movie, a three-dimensional movie. Here, this is shown as a projection in 2 dimensions of whole brain neural activity as animals are maybe thinking or deciding things or observing the world or producing actions. So here you can see a top projection of about 110,000 neurons at single cell resolution in this animal. And so that's the work that we did with the people I mentioned earlier, and that produced three-dimensional movies. So this is a movie like you just saw, but now it's projected along the 3 different axes: the front, the top, and from the side. You can see individual neurons lighting up, and so this represents neural activity. And in humans, this is kind of what makes us talk, perceive the world, walk, and so on. So in terms of numbers, we get about 120,000 neurons, 90% at single cell resolution, and we can get about 5 volumes per second. Of course, the speed of thought is even faster than that. Ideally, we would have millisecond resolution, but this is already good enough to have a summary of what's happening in the brain of this animal. Here's some more technicalities. This was an early implementation of this system where the fish was pointing up, and we had 2 light sheets from the side and 1 detection objective from the orthogonal direction. We've since modernized it, so now the animal is sitting horizontal as if it were actually swimming. We have orthogonal light sheets and so on. So this system has undergone many developments since its first instantiation. But this was an early, just because I want to cover some of that original paper that got the award. These were some of the networks that we discovered. So these are coordinated sets of neurons in the brain and their activity patterns. And since then, we've discovered much more about what these cells are actually good for. It turns out that some of these neurons are involved in exploration. So as animals explore their environment in search of food, for example, that's being controlled by some of these networks that you can see here, which were computationally extracted from these whole brain imaging movies. So one way in which we continued technology development is by creating a virtual reality environment for these animals, so they can swim around in 2-dimensional virtual reality environments while their brains are being scanned, as I just described. Now, the way we capture behavior is through electrical recordings from the tail, kind of like EEG recordings in humans. So we can tell through electrical signals that the muscles that the animal is trying to swim. So here you can see about 6 seconds of data with a couple of swim bouts here. It's swimming, pausing, swimming, pausing. So it tends to swim about once per second when it's swimming. It's often also resting or turning. But these are the signals we get from these nerve recordings. We can then decode these and feed them back into a virtual reality environment that we can project underneath the fish, so that the fish actually has the sensation that it's swimming through an environment. It's kind of similar to when we play a computer game, when we also see a projection of a virtual reality environment. That's exactly what this animal is experiencing. And we base the control of this environment on these electrically recorded swim bouts. This is a representation of a forward swim bout, and this is a turn to the left. So we are these systems are now quite good at decoding what the animal is trying to do and feeding that back into motion in VR. And we can combine that then with one photon, in this case, light sheet imaging. Here you can see such an environment. The fish is here. It's small and transparent. It's hard to see, but you can see the electrodes recording from it, and it's swimming around. It's swimming around in 2 dimensions. against a simulated water current. So when the fish is not swimming, the environment is moving as if the animal is being moved along with a water current, and then the animal can turn and swim against this current to stabilize its position in space. And I'll get back to this particular behavior very soon. So maybe I'll pause quickly, see if there are any questions.
Neural connections as well as just the whether they're on or off.
Yes, so that we're getting it. It's a field called connectomics. So the connectivity is very difficult to analyze because these are super dense. You know, it's like a ball of spaghetti except the size of a building that you have to use electron microscopy for and machine learning to track all these. But that's happening. So we will have it, but we don't have the full connectivity diagram yet. Thanks.
Maybe a naive question. You're saying it is swimming, but then it is fixed.
Exactly. Yeah, it's fixed. It's fixed so that we can more easily image from its brain, right? So it is like you're sitting playing a computer game. You're not moving in your chair, but you have the sensation that you're. It's in a. Yeah, so its head is encapsulated, or actually its neck. There's like a bridge of what's called agarose. It's a kind of jelly which hardens a little bit. It's still flexible, but it's hard enough to keep the animal in place. And when they're in that situation, if they're in virtual reality, they behave quite naturally. So this, yeah, we were lucky that this worked because at the start we didn't know if animals would do that because it's a little unnatural, right? But it works. It was based on work that people have done in mouse and in insects, where it also works fairly well. Oh, you mean in a mouse because they have a hard skull? That's true. Yeah, you got that right. So here it's a little harder. We had to work harder to make this work. Yeah, we tried all kinds of things. Yeah. So then after that, we continued technology development so that we had VR behavior during whole brain imaging. We coupled this now to online large-scale data analysis with cluster computing. So in the institute, there's a compute cluster. We could very quickly pipe the data from the experimental setup to the compute cluster to perform things like clustering, regression, and other types of statistical or machine learning analyses. And then based on the analysis, so here you can see the movie, the activity movie, and there you see a static volume where now the color represents aspects of neural activity, like is it tuned to visual motion, is it involved in generating swimming, and so on. So we can then use these analyzed volumes and combine it with cell perturbation methods. So you can use, again, molecular tools in combination with light to activate or silence neurons. And so with the human interface, you can then couple the two let me see if I can play this movie. No. You can couple the two so that based on the analyzed data, a human or potentially an algorithm can decide which neurons are going to be activated or silenced to see how that affects whole brain activity and behavior. So you can do causality tests and model refinement all in the same experiment. And so this is sort of an introduction to the system that we're using. And now I would like to walk you through a study we performed using all these systems on how the animal's brain encodes space. And there have been many studies on, for example, place cells in the hippocampus in mammals, finding that there are cells in the hippocampus that represent where an animal is in a room, for example. More recently, it's been also found by the Rowley lab that zebrafish may have such cells in the forebrain. This is about some other representation of space that I will describe in the context of this behavior that is quite well known across fish species. So here you can see a fish. This is not a zebrafish. This is just a random fish that a collaborator found in a river near the Genelia research campus. And it's swimming against water flow. You can see that, right? It's the water wants to move it to the right and the bottom of the screen, but the fish is swimming to the left and the top of the screen to stabilize its location. And this behavior makes sense because if animals wouldn't do it, they would just always move along with water flow and potentially end up in dangerous areas, or maybe on dry land if there's a drain, if a body of water is draining. So we call this positional homeostasis. Here's a cartoon of it. Larval zebrafish also do this fairly reliably, although I should say they don't always do it. It's just that their behavior is biased towards doing this. They can abandon it, go and hunt, or interact with other animals, but on average, they stabilize their location in space. And so we call this positional homeostasis. It's also known as the optomotor response or rheotaxis, but we call it positional homeostasis to emphasize the fact that animals maintain approximately their position in space. And we wanted to know how they do it. So it's very it looks very simple. You see yourself move and you swim in the opposite direction. But there are multiple ways in which it can actually happen. So one of the ways is a simple stimulus response algorithm where it doesn't require any memory. And the way you would implement it is if the animal senses a flow, it simply performs a swim bout in the opposite direction. And this will approximately work, but it probably wouldn't work very well. For example, if the animal wants to hunt for a few seconds or a few minutes, it would be moved along with water flow. So another, perhaps better way of doing it is through path integration, and this would require a memory of displacement and a way to add vectors to each other. So here you can imagine as animals are moved along by the water flow, maybe in 2 dimensions or even 3, every moment they integrate and add these vectors to provide an estimate of the global displacement at some future point in time. And then at some future point in time, they may be able to invert that vector and swim back to their original location. And then there's also a possibility that they don't use any of these, that they simply use landmark recognition. Maybe they see that they're close to a certain plant or a rock, and they just kind of create a memory of that image and stay close to it. So we wanted to know which ones of these are at play, and we're especially interested in memory in this animal. I mean, that's a personal interest of mine. I'm very interested in how the past influences the future through memory in the brain. So we wanted to know what are the multi-regional mechanisms, is there memory involved, and where is this memory stored? And this is work by N. Young, an extremely talented postdoc who's now a professor at the University of North Carolina, Chapel Hill, together with collaborators Martin Swart, Mikhail Rubinov, Zicheng Wei, Benjamin James, and James Fitzgerald. So let's see if we can test these different algorithms. And I know this sounds very simple, but if you really drill down into animal behavior, you find that seemingly simple behaviors are often not that simple once you really start understanding them. So let's see if we can test the stimulus response model where there's a flow in one direction and a swim in the other direction. So this would be a control theoretic implementation, a variant of a PID controller where there's water flow velocity, which pushes the fish in the environment, turns into fish velocity, which the fish perceives through optic flow. So the velocity of the visual scene, which is opposite to the velocity of the fish. And that then leads to corrective swimming, which then influences fish velocity. So this is the very simple way of implementing this. And the way we could test whether this is true is by creating an assay or a behavioral paradigm where we have a fish in an environment with a pattern. So given the pattern, the fish can see the environment move. And here I'm drawing this dashed horizontal line just for our reference. This is the starting point. And imagine there's water flow that pushes the fish backward. The basic behavior that we've seen this video of the animal in the river perform is that the fish simply swims kind of against the flow and will approximately stabilize their location in space. But now if we perturb this by kind of providing an impulse to the fish before water flow, by pushing the fish forward and then waiting several seconds and then switching on the water flow, the fish would simply perform the same behavior but elsewhere in space. And so without memory, under the simple stimulus response implementation, it wouldn't be able to correct for these perturbations. And so if we do this multiple times in virtual reality, then we can see the animal wouldn't be able to correct for these perturbations that have occurred in the past. And these trajectories would not converge. Of course, we would do this all in virtual reality because it would be difficult to actually push the fish, although it's possible, but in virtual reality, we can push the fish by just moving the virtual reality environment underneath it in an open loop way, so the fish doesn't have control over it. We just shift the visual image underneath. So that's one way. If we see this behavior, we would conclude, yep, they don't have memory. It's a very simple response. On the other hand, if we want to test whether they're performing path integration, we can we expect to see something different. So that would correspond to a PID type controller where there is not only a representation of velocity, but also a representation of integrated velocity or displacement of the fish. And if you have these two terms, then animals should be able to path integrate. And we can test this, again, let's simplify to one dimension. In this case, if we provide a perturbation to the fish, wait for multiple seconds, and then switch on this virtual water flow, the animal should take the prior displacement into account. Maybe it waits longer before swimming against the flow to get back to its original location, and vice versa. If we pull the fish backward, it might swim faster initially to return to its original location. And in this case, we would expect these trajectories to converge. And so through this very simple paradigm, we can determine which of the two behavioral algorithms are at play. And so we call this first period the pre-displacement period and the second period the swim period. And there's a delay period in between. The first two periods are under the experimenter's control. So in some way, we're asking the animal a question, hey, I just did something to you. And then during the swim period, that's under the fish's control. That's kind of the chance for the animal to respond, to say, hey, I remember you did something to me. I'm now going to correct for it. Or maybe not, or maybe they don't remember. And we loop this paradigm so we continue, so we can get multiple data points. We can simulate this as well because we have these PID models. If we simulate that, then you can see if you perturb in the model, you perturb them forward or backwards, and then have the delay period and then the swim period. So this is the model. You can see that these trajectories remain separated. Namely, the model has no memory. But if you provide the model with this integral of velocity, then the model can correct, and these trajectories will converge. And so here now is the result that N obtained, and this is a beautifully clear result. It's a super clear confirmation of the second model. So the animals can integrate over space. Here the little fish is not drawn to scale. We move the fish by about 10 body lengths forward or backward. So this is a substantial displacement. And if you look at the time axis here, this is 5 seconds. The time of convergence is about 15 seconds after the original displacement. So from this, we also know that the memory after 15 seconds is still almost perfect, because you can see that the green and magenta traces and the blue trace are almost perfectly aligned. Using additional experiments, we could establish that the memory lasts for over a minute, which I think, for us, it's not a big deal. Of course, we can remember things that happened a minute ago. But remember, the brain of this animal is so small. It's between 500 and 5,000 times smaller than the brain of a mouse, depending on how you count. 500 times the number of 500 times less neurons, 5,000 times less volume than the brain of a mouse, and the brain of a mouse is already small. So the fact that this tiny brain can do this is, I think, remarkable. Because it's tiny, it allows us a way in to try and understand how this works. So this seems to be the correct model. the PID controller, or the PI controller, the proportional and the integral term. And we were especially interested in this integral term because I think memory of the past is very interesting. Because I think the way that the past determines our future is one of kind of the core pieces of being human or animal. All right, so we established this path integration. Any other questions? I need to hurry up, I realize. Okay, so now we want to identify the underlying brain mechanisms. Here we turn to whole brain imaging during this behavior, cellular resolution, so we can examine what individual neurons are doing and look for this displacement term. And now, because of the way that the assay is set up, we can do this fairly easily. Of course, understanding the brain is tremendously complicated, but if you have such a clear assay and a clear behavior, you can sometimes find things by doing a very simple analysis. And so here we simply took the ranked correlation between the location of the fish in virtual reality and neural activity, and got, this is a combined map across about 6 fish, specific brain regions coding for position. So from these brain regions, you can tell, you can read off where the animal is in space. And these are specifically localized brain areas. So this is all single cell resolution data. It looks a little bit blurry because there are multiple brains combined here, and within these combined brains, there are small anatomical differences. But we see specific brain areas like area slow-mo, which we termed for self-location encoding medulla oblongata neurons, the inferior olive. So we termed slow-mo ourselves because that's kind of an unknown brain area, although we think it might be the nucleus purpositus in mammals. It doesn't matter what these names are, but it hasn't been specifically named in zebrafish, so we gave it our own name for the time being. The inferior olive is very well known. It's involved in motor learning and movement. And the cerebellum, similarly, is very well known, also involved in motor learning and movement. And then the dorsal raphe nucleus and the interpeduncular nucleus. This is interesting because the interpeduncular nucleus has been discovered by Ruben Portugues' lab to be involved also in angular navigation, and then the habenula. So these are all candidates now for implementing this location memory. We know from anatomical tracing and from the type of electron microscopy I was referring to earlier that the area slow-mo and the inferior olive are connected, and we know that the inferior olive and the cerebellum are connected based on decades of research. So we're starting to get to a multi-regional candidate mechanism for this. And of course, this is super coarse here, and an entire region is just drawn as a blob, so don't take this too seriously. We're just talking about now the very coarse connections between brain areas. And we can start looking at slow-mo, the inferior olive, and the cerebellum as initial regions just to see how does neural activity in these areas encode the location of animals. And here we can see something very interesting in area slow-mo, which was kind of the biggest area that coded for the location of the animal. Here we see 2 types of neurons that are spatially segregated. And if we look at activity on the cases where the animal moves forward and then swims back, or moves backward and then swims back, we see that neurons here code for these displacements, but they code for it in kind of an interesting way. So this is an example cell. It's one of those magenta cells. This codes for location by either increasing or decreasing its firing rates, its activity. Here, black is when there's no motion at all, when the fish remains stationary. But in addition to coding for location through increases or decreases in activity, they also show ramping activity over time. So in some sense, they also encode the time since they last were swimming. So when they swim, activity goes down. When they don't swim, activity starts going up, but it's mixed with a location signal. There's another neuron here, and another example, which has a much more pure representation of the fish's location. Here, if the activity is high, the animal tends to have moved backward, and if activity is low, the animal tends to have moved forward. So there's a variety of ways in which neurons can encode location. But in all of these cases, they're performing positional integration because a short, less than 1 second velocity stimulus leads to a long-lasting change in neural activity. So these are all integrators. And if you now want to look at activity across the population, ask what information is contained in this group of neurons, you can do network embedding, where you take activity from several hundreds of neurons and you reduce it down to 3 numbers and you represent that in 3 dimensions. So there are many ways to do that. In this case, we took one of the simplest one, principal component analysis, and you can see that depending on where the fish is, you get different trajectories. So for example, here, if the fish moves backward and then swims forward, the network trajectory goes through this left arm, and if the fish moves forward and then returns to its original location, will go through the rightmost arm. And we also found that this occurs in 2 dimensions and also in complex or random displacements. So instead of just moving the fish forward or backward, we wiggle it randomly forward or backward. It ends up in a random point in space. We can still decode from this population where the animal is in space. Any questions? All right, so this was all kind of a correlational analysis. We looked at what the brain is doing. We had some ideas of what that information was for. But we can now ask the tougher question. Is this just representing what's happening to the animal, or is this actually causal to the behavior? There are many cases where brain areas may represent the location of the animal, but the animal doesn't actually need that activity to have the memory or to swim back to its original location. So we wanted to know which areas were actually mechanistically implementing the memory and driving the animal to return to its original location. So we can use silencing and activation experiments to test this. So one thing one can do with a powerful laser is to focus it on a single cell and you ablate that cell. So you kill an individual neuron. You can do that multiple times. You can remove a set of neurons from the brain. And we can compare that to models. So here we have the PID controller, and we can ask the behavior before ablation. Well, we saw that before. So this is model, this is simulated. Now if we remove this displacement term after this kind of model ablation, the animal doesn't, these trajectories don't converge anymore as you saw before. We can now do the same thing in the fish. So now this is the real fish, which qualitatively looks like the model with memory. So this is an intact fish. It has memory. That's why the trajectories converge. If we now remove this area slow-mo, then this fish also loses its positional memory. And I think what's remarkable here is that it loses 100% of the memory. It still swims, but it has 0 memory left of where it was before. We can then also try to perturb the memory. So this is kind of a manifold representation of what we're doing. So think of this PCA space. We're trying to push the direction, push activity in this direction or that direction, and seeing if we can artificially implant memories and make the fish think it's moved, even though it hasn't actually moved. So this was technically not so easy, but here's an example where N first performed recordings of this population of neurons and found the neurons that code for forward or backward movement, and then used a technique called optogenetics, which is a combination of molecular biology and light delivery, to very specifically activate neurons that encode the memory of forward displacement or the memory of backward displacement. And she found that she can implant artificial memories that are demonstrated here. So in purple, for example, she stimulated the forward cells, and you can see at the trace below, the fish position at time 0 goes down. Namely, the fish waits longer before swimming. And then conversely, if you stimulate the backward cells, you trigger the memory of backward motion, and the fish swims harder. So in some sense, you've fooled the fish into thinking that this happened, even though it never actually happened. And so this is a very strong causality test, that this is indeed the locus, the implementation of this displacement memory. we think that humans may have similar displacement memories in the same kind of homologous brain area in our own brainstem that we may also use as we're walking around the environment. And this is something that still needs to be tested in mice, maybe eventually in humans, who knows. But this is still future work, hopefully to be done in collaboration with labs that are focused on mouse research. So just to wrap up, we also found these positional representations in the inferior olive and the cerebellum. Sorry, the connecting lines are switched, but when you have this multi-regional network, information is passed from one region to the other. And so here you can see that the inferior olive is receiving this displacement signal from area slow-mo, and then the cerebellum is receiving this displacement signal from the inferior olive. But each brain area has its own specific connectivity and does its own computations on these signals, eventually leading to behavior. So in this way, because we have whole brain activity, we can track how information travels across the brain, how representations of the environment or past events or motor planning and so on, decisions, how these are morphed between areas en route to trying to understand whole brain computation. And just a view forward to the field, I think it will be very fruitful to combine our understanding of algorithms underlying behavior with whole brain activity and connectomics to build detailed circuit models together with kind of a more abstract understanding of neural activity. And one thing that would really help is to have combined functional imaging and connectomics to bridge, as you said earlier, to bridge activity and connectivity. Another thing we're intrigued by is the contribution from the other parts of the animal, not just the brain. As I said to you before, those wild dogs were probably initially attracted to the gazelles because of hunger. That's a signal that originates in the body. So we're also looking with Virginia Rutten especially at the tissue outside of the brain to understand how signals travel from the brain to the body and back, and to also include these body-wide networks into a conceptual understanding of biological computation. In general, I think, I'm fond of, I originally came from mathematics and theoretical physics, transitioned to neuroscience because I think it's exciting how the field kind of gets input from a lot of different fields, from control theory and microscopy to physiology and cell biology. So also, if anyone, I know this is kind of a conference centered on mathematics and physics. If anyone's interested in talking to me about this, I would be happy to chat. Again, thanks to the collaborators and co-authors on the original manuscript that brought me here, and thanks to all the other people who were involved in this research, and thanks to the organization again, and thanks to you for attending. Thank you.
So, any questions? Okay, maybe I will have a little question. Like, you use laser to kill those neuron cells. Is it possible to use some chemical to block this neuron connection? And then, after sometimes this neural connection is restored, and then this fish will behave like normal?
Yeah, it's a great question. It's very possible. The one challenge with chemicals is that the chemicals don't have a notion of which cell to go to, right? So it might affect the whole brain. But there are techniques where you can combine proteins with designed chemicals so that you can express a certain protein in a specific cell type. The chemical will then specifically go to that cell and change that cell, and then you can wash it out. So yeah, it's a more complicated experiment, but it's very powerful because it's reversible.
Yeah, but so far I don't know. But the laser can kill that your designed cell type. Is that true? Okay.
Yeah. The laser. So typically, what we do is to image from the brain and then to quantify this cell, this cell encodes that stimulus or this movement or this action, and then specifically target that one. Yeah, there are also other ways to do it. You can use molecular biology with light to silence the cells. So there are even photo-switchable proteins where you can use one wavelength to silence the cell, another wavelength to switch it back to its normal state. So you're right. There are more elaborate and more precise ways of doing it. The question is always, how complicated does the experiment become? Do you need another microscope and another person and another month or longer to do that experiment? Or yeah, so we usually try to keep it simple, but of course sometimes we need that.
When you kill, I guess you did try this, but if you killed off those neurons with the laser, and then maybe you let the fish go for a few days and do the same experiment again, does it recover its ability? Or?
It's a very nice question. So in some cases, yes. In some cases, no. And in some cases, something else happens. And this was actually a very interesting case where if you kill those neurons, there are other neurons that kind of pop up. And we think that the network, the slow-mo network we focused on, were actually inhibitory neurons that are reciprocally connected. So if you kill one of them, then there are other cells that were previously suppressed by the one you killed that then become disinhibited and start showing activity. And we think that's got something, we think that has a strong connection to the computation that this region is actually performing. We have an entire model that we never published for, I don't know why. We should do it, but it's kind of unfinished, where we have a model of how these cells are connected in chains, and where you feed in sensory information, and the memory arises because information propagates along the chain on slower and slower time scales. And in this model, that sort of that has that property, that because there's mutual inhibition, you get these. Okay, so that's in this case. In other cases, there are cells that when you kill them, they don't really come back. But especially because this is a developing organism, there are many cases where you can take out a huge amount of the brain and it grows back. And it's very interesting that humans don't have that property, but certain fish do have that property, and we don't know exactly why one species can do it and another species can't. I wish humans could do it, obviously. Thank you.
Yeah, very nice talk. I don't know much about it, but it was really interesting. I have two questions. One is the generalizability of your analysis to different fish, maybe. Like, do you do it on one fish and just analyze, or the same networks are activating on all the larvae you see, or under different environment, etc.? I'm just curious about that.
Also a great question. So the observations in the analyses you saw are very generalizable across animals. So there are differences between animals, but the major differences are already visible from behavior. So some animal, a single pair of adults gets maybe 300, and one mating event can get 300 offspring. And so not all of them are kind of good, and some of them may not perform these visual behaviors very well. In those cases, you see differences in brain activity. But in the animals that perform these visual behaviors well, and I do think this is kind of, those are the ones that would survive in the wild. It's very consistent. These brain activity patterns are very consistent. But of course, there are individual differences, and it's also possible that there are, this is one behavior. There may be other behaviors, other challenges that animals solve in a way that is very individualistic. And in those cases, you'd want to find a way to kind of jointly analyze different individuals within the same framework. So again, a project that's kind of been ongoing, but that's now stalled because the first author moved to a machine learning company and is now earning so much money that he's kind of lost interest in neuroscience, but as sometimes happens. But a machine learning framework for creating kind of I guess you would call it a foundational model of a fish brain that then has specific instantiations in different animals. So that's something that, a line of work I hope will make progress in as well.
The other thing, like following up on that, how the fish is held in place. So I understand, I guess, if it is moving, it is difficult to do light sheet microscopy or any imaging maybe. But would the behavior change? Because you're calling it swimming, but I don't know if it is swimming. It is like, it is trying to do that action, but
Yeah. So
there will be some other feedback that it may get when it is moving in the water. And
so we always try to validate things we see in VR with things we see in free-swimming fish. And these animals do this behavior when they're freely swimming. But from what you said earlier, it is actually possible to image in free-swimming fish. So two of the collaborators at the Rowley lab I mentioned earlier, they've created the technique for imaging in free-swimming fish. And then Kai Wang and Chen Wen at Kai Wang is at ION in Shanghai, developed also a method for imaging in free-swimming fish based on computational microscopy, light field imaging. It has its own set of challenges, but it is possible, and I think that's a very fruitful. It comes with you have to register that the brain is constantly shifting, and but it is possible, and I think yeah, it's always. I mean, eventually we would love to go to India or places where they live natively, build microscopes in the wild, but that's probably impossible, at least for a few more years. But yeah, neuroscience is often a compromise. But I think that as a field, we're aware of the need to go to more and more naturalistic settings.
Any more questions?
I have a simple question. Did you use independent components analysis to analyze the data instead of the principal components? Maybe it is better.
Yeah, I'm pretty sure we have done that. We tried a number of different approaches. So, but ICA is not sort of the most used in the lab. We often use PCA, NMF, non-negative matrix factorization, or factor-based clustering. So factor analysis followed by a rotation of the axes to minimize the overlap between the factors. But this is it's a big field in neuroscience, dimensionality reduction, or more compact representation with large numbers of neurons. There are many variants, and I think a lot of progress is needed from mathematically oriented people to understand population dynamics. Thanks. Thanks for the great questions.
Any more questions? Let's thank our speaker again.
Thank you.